and cell therapy updates, covering the latest breakthroughs. Brought to you by Lucid Quest. Professor Ayel Hendel of Bar Ilan University has joined the EU-funded EASYGEN Consortium, a 5-year, €8 million initiative to improve hospital-based CAR-T therapy delivery. The goal is fully automated point-of-care manufacturing that provides CAR-T cells to patients in about 24 hours. Hendel's lab will apply CRISPR editing and off-target safety analysis across the workflow. In Saudi Arabia, the Saudi Food and Drug Authority has approved its first Phase 1 trial of a locally developed CAR-T therapy for CD19-positive acute lymphoblastic leukemia in adults aged 18 to 60. Developed at King Faisal Specialist Hospital with lentigen and miltenyi, the cells will be made in-hospital using a closed transduction system. The trial focuses on safety and aligns with Vision 2030's health innovation goals. In bladder cancer, Phase 2 data for detalimogene voraplasmid, a non-viral plasmid-based gene therapy, were released from the LEGEND trial. Results show a 71% complete response rate, with 51% maintaining response at 6 months. Response fell from 67% at 3 months to 47% at 6. The shelf-stable, non-integrating therapy showed mostly mild local adverse events and no systemic immune-related toxicity. Fresenius Kabi and Charles River Laboratories have joined EASYGEN to support hospital-based CAR-T manufacturing across Europe. Backed by €8 million from the Innovative Health Initiative, the aim is to expand access, as treatment rates in the EU remain under 20%. The platform includes Fresenius's cell manufacturing, Charles River's 3D screening, and Celix's Einisch analyzer for safety and quality checks. Vyriad and the Weizmann Institute have signed an exclusive license for a novel in vivo gene delivery platform. The technology uses adapter peptides to mask viral receptors and redirect viruses without altering genetics. This preserves potency while improving targeting and avoids complications from envelope mutations. Vyriad will apply it across viral and nanoparticle programs, including CAR-T. Encoded Therapeutics received FDA RMAT designation for ETX-101. It's AAV9-based gene regulation therapy for SCN1A-positive Dravet syndrome. Early Phase 1, Phase 2 data suggests seizure reduction. The Polaris trial aims to complete enrollment in Q4 2025, with interim data expected the same quarter. The company's GMP site in North Carolina is producing clinical supply to support pivotal trials in early 2026. Hope Biosciences also received FDA RMAT designation for HB-AD-MSCs. It's stem cell therapy for relapsing remitting multiple sclerosis. A 24-patient Phase 2 trial with 6 infusions totaling 1.2 billion cells met its primary endpoint. Patients showed significant improvement on the MSQOL-54 scale. Hope is preparing for accelerated approval pathways, including a BLA and manufacturing readiness pilot. Kyverna Therapeutics provided updates on KYV-10H1, its CD19 CAR-T therapy for myasthenia gravis and stiff person syndrome. The KYSA6 trial will enroll about 60 patients in a Phase 2, Phase 3 design, randomizing 1:1 to KYV-101 or standard care. Co-primary endpoints are MG-ADL and QMG at 24 weeks, with crossover allowed. Phase 3. Enrollment is expected by late 2025, with interim Phase 2 data in Q4. Finally, a study in Frontiers in Pharmacology explored gene therapy preferences in Duchenne muscular dystrophy. Based on interviews and literature, 48 influencing factors were identified. 6 key attributes emerged: therapy type, impact on life expectancy, risk of severe events, delay in ventilatory support, years of functional preservation, and depth of supporting data. Caregivers of younger children showed higher interest in gene therapy trials. Stay ahead in cell and gene therapy research. Like, share, and subscribe for weekly updates. Visit lqventures.com or email us at info@lqventures.com.